Hello, everyone, and welcome to today's webinar sponsored by AnaBios Corporation. My name is Chris Mathis, and I'm the Chief Commercial Officer at AnaBios, and it's my pleasure to be the moderator for today's session. Before I introduce our speaker, I'd just like to say a few words about AnaBios. Many of you on the webinar today in the audience know AnaBios, but for those of you that don't know us, AnaBios is a unique contract research organization and biotech company based in San Diego, California. We recover human organs from ethically consented donors and use the tissue or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We've essentially redefined first in human. And working with AnaBios as a partner is as close as you can get to a clinical trial without actually doing a clinical trial. So for more details, check out our website at anabios.com. So just a few reminders, today's session will be recorded. So if you miss a detail and would like to go back, you can do that on demand. And we will be taking questions at the end of the webinar. In your GoToMeeting or your GoToWebinar panel on the bottom right corner, you'll see a questions box. That's where you can ask questions. So it's my pleasure to introduce our speaker today, Dr. William Renthal, who I learned has been a collaborator of Clifford Wolf, who many of you know from the pain field for about 3 years. And he met Dr. Wolf while doing his residency. So Dr. Renthal is presently a Director of Research at the John R. Graham Headache Center at Brigham and Women's Hospital. He's also Assistant Professor of Neurology at Harvard Medical School. He completed his MD, PhD, and neurology residency at the University of Texas Southwestern Medical Center and fellowship training in headache medicine at Brigham and Women's Hospital. Dr. Renthal treats patients with refractory headache disorders and directs an NIH-funded research laboratory at Harvard that studies the genetic and epigenetic mechanisms underlying headache and pain disorders. His work uses cutting-edge genomic and neuroscience techniques to identify new strategies for developing headache and pain treatments. He recently received the Career Award in Medical Sciences from the Burroughs Wellcome Fund and the American Academy of Neurology Harold Wolff John Graham Research Award. And he has published over 40 peer-reviewed articles and book chapters. So again, it's my pleasure to introduce our speaker. The title of his presentation today is Flexible Identity of Peripheral Sensory Neurons. Dr. Renthal, take it away.
Thank you so much for that introduction. I'm really looking forward to the opportunity to share with you all some of the work we've recently published and some ongoing studies in the lab. And hopefully there'll be some time left at the end to talk about ways to extend these findings and even possible opportunities for collaboration. These are my disclosures. So as a headache and pain neurologist, I think it's really important to highlight that pain, migraine, and then opioid-related complications of these conditions represent approximately 1/4 of all years lived with disability across all conditions. So when we think about this from a population level, there is really a massive return on investment in terms of quality of life if we can develop better ways to treat these and manage these conditions. Obviously, because of the prevalence of these conditions, these conditions exert a pretty tremendous toll economically, with over $600 billion for pain per year for direct and indirect medical costs, and then $40 billion per year in migraine, and these are numbers that are going up. In addition to the economic costs, more importantly, these conditions often require opioids for management, and most of us are aware, over the past decade, opioid-related overdose deaths have doubled. And this is largely a consequence of the off-target actions of opioids on the neurons that control reward and respiration. And so there's really been an urgent need for the development of next-generation pain therapeutics that are highly specific to the neurons involved in pain processing without these off-target actions that are seen with opioids. So pain. So the pain system has been studied for a very long time and involves a number of different neurons that exist from the periphery, where the actual sensory stimulus is converted from a change in temperature or pressure into an electrical signal that's transmitted to the dorsal horn of the spinal cord through these peripheral sensory neurons. There in the spinal cord, these signals are integrated and transmitted rostrally up to the brainstem and then into the somatosensory cortex, where we think the perception of pain occurs. And you can imagine that new pain therapeutics could really target any one of these neurons along the way, if we can understand enough about them to develop specific therapeutic interventions. And indeed, there are a number of really great labs around the world that are focusing at each step along the way. The work I'm going to talk about today is going to focus primarily on the peripheral sensory neuron, although we'll touch a little bit on the spinal cord at the very end. And the reason why I think this is a really important place to begin is because most forms of neuropathic pain at some point converge on this cell type. So zooming in on the dorsal root ganglia, which houses these peripheral sensory neurons, the cell bodies of these peripheral sensory neurons, you can see here that they come in many different shapes, sizes from this cartoon, and even colors to some extent. And they're highly specialized neurons that can detect distinct sensory stimuli, as well as project to different places in the body. And so one of the many ways these neurons are different, in addition to the size, in addition to the place they project to and how thick they are, whether they're myelinated or not, whether they secrete peptides or not, are the transcriptional diversity within these cells. And if you can think about the unique features that are perhaps within the neurons that transmit pain signals, or classically termed nociceptors, we might be able to identify new therapeutic strategies to more selectively gain access to and control these cells in pain conditions. I think one limitation of the field over the last 20 years or so that people have had this same idea has been that analyzing these tissues in bulk, along with all of the glia and the different subtypes of neurons within this region, has been very difficult, and to identify unique molecular features. And so recent advances in single cell genomics have now made it possible to understand some of these molecular features within specific subtypes, as well as how they respond to noxious stimuli that are known to cause neuropathic pain. And so as I alluded to earlier, that many forms of neuropathic pain, many causes of neuropathic pain converge on peripheral sensory neurons, ranging from things like chemotherapy-induced neuropathies, viral infections like zoster, and the one I'm going to talk about today is a very common condition that occurs after surgery or injury, where people can develop long-term chronic pain after these events for reasons that are not immediately clear. And so I'm going to spend the rest of the talk talking about peripheral nerve injury as a model system for understanding neuropathic pain. One of the things that appealed to me, I'm somewhat of a newcomer to the pain field, but one of the things that really appealed to me about nerve injury is beautifully illustrated here in a video from Alvaro Sagasti's group about a little over a decade ago, where they take a cultured trigeminal nerve or neuron and shine a laser to destroy its axons. And I'm just going to play a video of what happens. Most of you probably know, but what occurs immediately after the laser is there's Wallerian degeneration of the distal axonal stump, and then regeneration of the axon. And to me, this is a really remarkable, I'll play it one more time, remarkable thing, because this cell goes from transmitting sensory information to, well, in this case, a petri dish, but imagine in vivo, transmitting sensory information to the spinal cord in 1 second. And then after this laser beam hits or the injury occurs, the cell changes states immediately into a cell that's focused on regenerating these axon terminals and reinnervation ultimately. And so I'm certainly not the first person to be amazed by this process. There have been many groups over the years that have studied the molecular events that occur after this injury occurs. And and it's been most recently, I think, beautifully reviewed by Valeria Cavalli's group at WashU, where after the axonal injury occurs, there's a back propagation of calcium that initiates an entire series of transcriptional events that are necessary for the proper regeneration of the neuron. And so this is really where I kind of have gotten really excited, because a number of these transcriptional events that have been described are transcription factors that are induced rapidly after nerve injury. And many of them are shown here, where perhaps the most famous of which is ATF3. But in addition to being upregulated after peripheral nerve injury, many of them are necessary for the regeneration. They've been shown through loss and gain of function studies, and some of them even are contribute to regeneration in central nervous system neurons. And so one of the questions when I started becoming interested in this that wasn't clear was where these changes were occurring and whether or not these transcription factors regulate similar or distinct transcriptional programs within these distinct cell types. And so when I was finishing up my postdoctoral fellowship with Mike Greenberg, where I developed some new single-cell RNA sequencing protocols for mouse and human central nervous system tissues, I reached out to a former advisor of mine when I was a resident, Clifford Wolf, to collaborate on trying to answer some of these questions. And so the work I'll be talking about today is a joint effort between our groups, in particular of Ivan Tuchitsky, a former postdoctoral fellow in Clifford's lab, and Light Yang, who's a research associate in mine. So the model that we're going to talk about today, or the models we're going to use to study nerve injury today are I'm going to talk about two, although we did several others that I'm happy to talk about in the question and answer session. I'm going to talk today about the spinal nerve transection and sciatic nerve crush. And the key difference that I want to highlight between these two is that in spinal nerve transection, it's a very proximal injury to the cell body, and because of that, we injure we injure the axons of most of the cell bodies within a specific DRG. Whereas in sciatic nerve crush, it's a more distal lesion, and axons branch off before then, so we end up only injuring about 50% of the cell bodies that we sequence in the DRG we collect. And one other key difference that occurs in our sciatic nerve crush model is that there's functional regeneration of these axons, and so that's another important feature. So what we do is, regardless of the model we study, we end up isolating nuclei and into a single nucleus suspension, and then using droplet-based single-cell genomics, in this case indrops, although we've used other methods as well. And the way this works for people who haven't seen this before is that we flow the nucleus at the very bottom here into a droplet that's contained along with a large molecular barcode that labels all of the RNA within a cell with a unique molecular barcode that we can then sequence and then trace back all the transcripts to the cells from which they came. We then sequence everything and then spend a lot of time analyzing these data sets. And the data we'll talk about today come from a little over 100,000 DRG nuclei. So to orient everybody to this, this is called a UMAP plot, where each dot on this plot represents an individual nucleus that we've sequenced. And the way this works is these plots take multidimensional data from the counts of every gene within each of these dots and clusters cells very nearby that are very similar in gene expression patterns, and pushes cells far apart that are very different. Nuclei in this case, very, very far apart if they have very different levels of transcription. And this occurs across thousands of transcripts. So unlike our traditional approaches where we look at an individual gene as a kind of a marker of a cell type, this has a much more refined view of what a cell type is based on the expression of thousands of genes. Now, again, these are transcriptionally defined cell types. These are not functionally defined cell types, although there's been some great work trying to correlate these from Alex Chesler and Nick Riva's group recently. And there's largely overlap, although some interesting differences. And so what I'm going to talk about here is I've assigned these clusters to specific functional cell types that have been described previously in the literature based on known marker genes. But the clustering is driven by the many thousands of transcripts. So just to kind of highlight a few, these blue cells here are the non-peptidergic nociceptors that express MRGPRD3. These orange ones are peptidergic nociceptors that express CGRP and substance P. These yellowish ones we call a peptidergic subtype because in mice, these express substance P, but also TRPM8. In the human, it seems like there's really not much peptide expression in these cells. And these green cells are large diameter fibers that we call a mix of 8 beta LTMRs and proprioceptors and A delta. So we were able to recapitulate most of the known DRG cell types, and these data are largely consistent with some of the single cell data sets that have been generated from Patrick Renthal's group and David Ginty, where they basically identify the same cell types, although using whole cell analysis tends to give better resolution of the large diameter fibers, whereas the single nucleus has a little bit less complexity of the transcripts we detect, and therefore the large diameter fibers seem to merge together more than with the whole cell data. So what I'm going to show you now is a time series of what happens to these cells after they get injured. Now, I'm going to now change the color of these to from the cell type to how much ATF3, which is an injury-induced transcription factor, is present in each of these dots. And so this is the naive condition, very little ATF3 is expressed. And then after 6 hours after spinal nerve injury, and again, this is the proximal lesion, you can see ATF3 is induced in these cells, but largely the cells look like their naive counterparts. They don't change the way they cluster. By 12 hours, roughly a similar perspective, much more ATF3 expression, but it's pretty similar clustering patterns. But by 1 day, you'll start to notice that these ATF3 positive cells start to move towards the center of this plot. And this continues after multiple time points and progresses to kind of a peak around 3 days, where almost all of the cells we sequence enter into this, what we call an injured transcriptional state, in which cells no longer cluster with their naive counterparts and express very high levels of these classic regeneration-associated genes. And this is we finalize this time course at 7 days after injury. So we can now quantify whether or not a cell is in this injured state, and then plot it as a function of time and get standard deviation over biological replicates. And we can see that the majority of these cells, as I just showed you, enter into this state within 1 day and peaks around 3 days. And it's important to note that we detect about 93% of the cells that we sequence are injured in this, enter into this injured state by 3 days, which approximates the number of axons that we physically, or the fraction of axons that we actually physically cut. So I think it's a pretty good model or a pretty good assay for identifying an injured state neuron or neuron that's been injured. I told you I would look at what happens after sciatic crush as well, a more distal lesion. And in this case, we injure about half of the neurons. And this is the time course we observed in terms of the fraction of neurons that enter into this injured cell state over the course of over 3 months. And so what you can see is that roughly similar kinetics, but it peaks around 50% because we injure only about half of those neurons. And then the recovery occurs pretty quickly between 7 days and 14 days. We're kind of regretting the fact we didn't do more time points in there, but it's work that's ongoing now. And then we see essentially a full recovery by 3 months. Important to note that the non-neuronal cells do not demonstrate this same transcriptional reinnervation. And that we also did a few other other models that have been known to cause neuropathic pain, such as paclitaxel and CFA, and neither of these induce the same injured cell state. This is something that's unique to axotomy. Let me rephrase that, at the doses that we use of these drugs. So we're doing ongoing studies, we're looking at higher doses of paclitaxel and CFA, and so we may have to revise that impression when we get those data. So that we sought out to answer questions that were, of what are unique transcriptional changes within distinct cell types. In order to do that, we have to identify what a cell type is both before it's injured and after it's injured, because we don't want to mix, let's say a CLTMR type neuron with after injury with a peptidergic neuron before injury, because you'll get a bunch of very cell type specific changes that are not related to the injury process. And so in order to do that, what we typically would do is take those injured state neurons and cluster them to find out which the different cell types they are, and then look for marker genes to confirm our bioinformatic analysis that the cell types are correct. And when we look at these marker genes that we classically use, what we find is that most of them are downregulated or not even detectable within a week after injury. And this makes it really complicated to identify what a cell type is after it gets injured, because these genes are all downregulated. And for the aficionados in the group, you know that single cell sequencing is not a perfect science yet in terms of the ability to detect every transcript in a cell. So we wanted to confirm that this wasn't just an artifact of sampling. And so we did in situs at for each of the marker genes, and I'm showing here a couple. If you look at in a naive DRG, TH or TAC1, the markers are in green, TUB3 and neuronal markers in blue, and ATF3 is in red. In the naive state, you see, in both 6 hours after spinal nerve transection, you see nice staining for the marker genes that we're looking at. But by 7 days after injury, we really see complete loss of these transcripts in most cases. And this is quantified here on the right. So the question is, how do we go about identifying what these injured state neurons are after injury so that we can compare them to their naive counterparts and do the type of analyses that we set out to do, which was cell type specific differential expression analysis. And so we ended up taking 3 complementary approaches. One is by developing a very high resolution time course that I just showed you. We were able to use a method that we developed called pairwise clustering and projection to project the cell type identities when they're known in the naive state to each consecutive time point, and then propagate those labels onto the next time point until we get all the cell types labeled. And I'm happy to talk about these in more detail after the talk. But the important point is that we used a separate bioinformatic approach that gave roughly the same results, and a cell type, a genetic label of these MRG-PRD positive cells to follow them after injury and make sure our bioinformatic analyses were consistent with the genetic label that we know is in non-peptidergic nociceptors. And with a grade of about an A-, we were able to identify all of these cell types with good accuracy. So this enabled us to now perform differential gene expression analysis for each cell type at each time point after injury. And this is a ton of data, and there's a lot of different things to look at in these data sets, and all of it is available on a website at painseq.com. If you're interested in specific cell types or specific sets of genes, you can go in there and graph the time points and specific parameters that you're interested in. But the key points I wanted to highlight on this slide are that neurons induce a lot of genes that are after injury, as you might expect, and non-neurons less so, although some. Perhaps a notable exception are endothelial cells that seem to be pretty active after injury. But the majority, and then one other point is that the kinetics of induction of these genes is slightly different between different cell types. And what you'll note is that these small diameter peptidergic and non-peptidergic neurons, or otherwise C fibers, tend to induce their gene expression patterns their genes after injury relatively quickly, within hours to a day, whereas the large diameter ALTMRs or A-beta LTMRs tend to induce gene expression much later in the time course. We haven't really dug in yet into the reasons for this, but that is one interesting observation that came out of it. Ultimately, the question we wanted to ask was how similar are these gene expression patterns between different cell types? So we ended up doing a pairwise overlap analysis between each of the sets of genes that are upregulated after spinal nerve transection. And what we found Basically, so in this case, just the way to interpret this plot is that these are the genes that CLTMR, this is the p-value of the overlap between the genes between CLTMR and CLTMR, which is nears 1 depending on the length of the list. I'm sorry, near 0 depending on the length of the list. And so here is what you'll see is that the more red the box in this heat map, the more significant the overlap is between the two sets of cells. And so you can see that the neurons induce largely the same sets of genes after injury, especially with comparison to the non-neuronal cell types. And so this suggested that there's a common gene program that may be overlapping between not only cell types, but perhaps even different cell types after injury, not only in the peripheral nervous system, but perhaps in other tissues as well. So one other way to look at this is based on which of these genes are actually cell type specific in terms of the fraction within each cell type. And the red bars here are the fraction of genes that are upregulated after injury that are truly cell type specific, whereas the yellow and green are the fraction of these genes within each cell type that are commonly shared across multiple cell types. And so you can see that the majority of the genes that are upregulated in any given cell type are these commonly induced genes. And so because of this, we became very interested in following up this common gene program. But one important point is that this injured state really is not just this set of genes that are upregulated after injury. And one important control to test this is if you look at the cells that are injured in this injured state, and then we cluster them, instead of based on their variable genes between all the different cell types, we cluster them without any injury genes at all. So we just take those out of the transcriptome. You still see that we don't get to identify that. We can't really identify what the cell types are clearly after they get injured. And this means that there's something else going on in these cells besides the upregulation of these hundreds of thousands genes after injury. There's something else going on that makes these cells very similar to each other. And so when we looked at this in a little bit more depth, what we found was that the genes that are typically define a cell type, meaning they're cell type specific genes, the ones that are different between different cell types in the naive state, are largely downregulated after injury. And you can see basically this because they're all, these pie charts are the fraction of these genes that are either up or downregulated. The majority of them are blue, which means they're downregulated. And this is actually the thing that most importantly drives the loss of cell type identity after injury. And so the mechanisms for this are still largely unknown. So, trying to get into trying to better understand the mechanisms that drive this what we termed injury-induced transcriptional reprogramming, we thought that we could maybe identify some of these features by looking at transcription factors that are upregulated very quickly after injury, and either some of them will persist and stay upregulated, some of them will go down. But if we identify the ones that are upregulated very early and then search for the transcription factor binding sites in the genes that are upregulated later on after injury, we might be able to understand some of the transcriptional network that's driving this reprogramming phenomenon. And so when we take a look at these genes that are upregulated, or the transcription factors that are upregulated very early after injury, who have some enrichment who have the transcription factors binding sites enriched to any degree at a later time point, we end up finding about 9 transcription factors. And many of these will be familiar to aficionados in the group, with ATF3 being one of the more prominent ones. And so to kind of help us prioritize which of these to start with, we did enrichment analysis of which of these factors had their motifs most enriched in the set of these commonly induced injury genes that we've been talking about. And what we find is that ATF3 and Jun, which can co-dimerize as an AP1 complex, are by far and away the most enriched transcription factors in the genes that are upregulated after injury. And so this led us to focus on this transcription factor more as a potential mediator of this injury-induced reprogramming phenomenon we observed. And so to do that, we generated ATF3 conditional knockout mice. and crossed them with VGLU2 Cre mice, which express Cre in most neurons. And we can see here is that here's both one week after, these are DRGs 1 week after injury, and the wild type mouse has nice ATF3 induction, whereas the ATF3 conditional knockout has really very little ATF3 induction. And that the ATF3 that is induced is from non-neuronal cells. And then to kind of recapitulate some of the findings that have been published previously, that ATF3 is important for nerve regeneration. In our conditional knockouts, you can see that using a pinprick score, which is just a function of the sensory response from these from the injured limb, you can see that the normal mice, or the wild type of the mutant mice, have very similar responses to pinprick at baseline, but then after injury. But the rate at which they regenerate their sensory improvement and restore normal functional sensory recovery is significantly delayed in the ATF3 conditional knockout compared to wild type. And so, given the fact that we were able to recapitulate this finding, we wanted to next understand, what's happening at a transcriptional level, and does this match what we see at a behavioral level? And so we performed single nucleus RNA sequencing in both naive and in mice that have had a sciatic nerve crush 7 days afterwards. And in this plot, you see wild type and ATF3 conditional knockout cells all mixed together. And the cell types have been labeled using the same approaches I talked about earlier. The pattern of this plot is different than the one I showed you before because it's a different experiment and a different set of animals. So the position on this plot is a little bit different, but the same cell types were observed. And what we find here is that we see a nice large cluster here of injured state cells and a smaller one up here that express high levels of ATF3 and are only observed in the injured animals, or not really found in any large degree in the naive animals. And so when we separate these out between wild type and conditional knockout cells, you can see that the injured cluster here that's now colored by an injury score, which I'll mention in a second, is largely present in the wild type animals and not the conditional knockouts. The ATF3 conditional knockouts have a very distinct cluster that looks like it has not really reached its normal transcriptional state that's observed in the wild type, and the injury score is much lower in these animals compared to the wild type cells. And so this injury score that I'm talking about is really just an aggregate score of all of the genes that are typically upregulated after injury in the wild type animal. So it's really notable that across this entire program, the ATF3 conditional knockouts are unable to induce this transcriptional state to an adequate degree. And so, I mean, it's clearly able to do so to change its transcriptional state a little bit after injury, but ATF3 is one of the key components. And so we're very interested in following up exactly how ATF3 is doing this. And also, what are some of the other factors that are involved in this process? Because ATF3 is not the only thing that's upregulated. And so to do this, we've begun a process of generating a CRISPR library of the key transcription factors that are expressed in DRG. And we've generated unique barcodes for each of these guide RNAs and are injecting this multiplex library into mice and then injuring their sciatic nerves or spinal nerves, and then we can, by single cell sequencing, identify which cells have which respective guide RNAs expressed in them based on the barcodes that they're associated with. And these experiments are still ongoing, so I unfortunately don't have the results yet, but just so you can kind of know what our thinking is, in our control guide RNAs that don't knock out any transcription factors and just maybe knock out a or just cause a double-strand break in an unimportant quote-unquote region, we would expect to see our typical injured state emerge after spinal nerve injury or sciatic nerve crush. Whereas if we knock out a transcription factor that's important for the generation of this injured cell state, like ATF3, we will see a reduction in the reduction in the number of cells that we see enter into this injured cell state. And so anyway, we're hoping to have a list of transcription factors that are necessary for this in relatively short order. So one of the questions that we've made a little bit more progress on, and I'm excited to share with you though, is how similar is this injury program in the peripheral nervous system to that in the central nervous system? And there are a variety of ways we can answer this question, but the most straightforward is to directly compare the motor neurons we're injuring after sciatic nerve or spinal nerve injury to those in the sensory ganglia. And so since we're cutting the same nerve, it seemed like a really good opportunity to take out the motor neurons that are they're also traveling in the same nerve sheath. The data I'll show you is from the following experiment, where we've taken a chat Cree mouse that expresses Cree in cholinergic motor neurons, crossed with a Sun1 GFP reporter that allows us to label the nuclei of any cell that expresses Cree. And we've performed sciatic nerve crush whether 1 day or 7 days after injury. And we took the lumbar enlargement or the ipsilateral lumbar enlargement, and then perform FACS to sort out the GFP positive cells. Then do 10x single nucleus RNA sequencing, and then analysis. And so what we see at this, this is just all of the data put together and the different cell types we see. We see are the main clusters that have been reported recently by Aaron Kittler's group, cholinergic interneurons, visceral motor neurons, and skeletal motor neurons, based on the marker genes I mentioned that Aaron Kittler's lab has recently put on by arXiv. And because we have a pretty similar overall clusters, what they've reported, we felt confident going forward in trying to identify what's happening in the skeletal after injury in these cell types. And when we just plot the whole thing together and we look at cells that express high levels of ATF3, and I'm not showing this to you, if you look at differences between the injury time point and naive time point, there's really only one cluster that stands out, and that is this cluster right here labeled in ATF3 that's present in the injured animals but not in the naive animals. And so we do see a very small but meaningful cluster of injured state cells. It was kind of exciting for us to see. So now we wanted to then understand the kinetics over which this injured state emerges. And what you can see here is that almost all of these cells, with a few exceptions, come from animals 1 day after crush. And by 7 days, this injured state has reverted back to its naive baseline. It's not to say there aren't some differences, but that there are not sufficient number of differences to really cause them to cluster separately. And this was a very different time course than what we saw in the dorsal root ganglia. So we're very interested in now understanding some of the similarities and differences for why these neurons tend to recover, quote unquote, quicker. We know that regeneration of motor neuron regeneration may happen maybe slightly faster, but they're still regenerating largely in these animals even at 7 days. So, excuse me, we think it's not entirely driven by functional reinnervation, but I'm happy to discuss this more in the question and answer session. Lastly, to just compare directly the peripheral nervous system and central nervous system regeneration programs, it's the genes that are upregulated after injury, either in the motor neurons or the dorsal root ganglia neurons. There's a really significant overlap. Just under half the genes that are upregulated after injury in the motor neurons are shared with those in the sensory neurons. So we're very interested now in wondering whether or not which other cell types share this same set of transcriptional responses. There are obviously some cell type specific differences, but this common set of of transcription factors may be particularly relevant for regenerative medicine, for instance. One last thing I wanted to share with you is that because of how similar this program is, we hypothesized that maybe ATF3 would participate in the regeneration of motor neurons after injury. And so we generated a chat-Cree ATF3 flox mouse where the Cre-negative mice are the wild-type ATF3, the Cre-positive are ATF3 knockouts, and then we have a couple of hets in this group. And what you see is after motor neuron injury, this is looking at digit-gait analysis, where the way a mouse walks is quantified over time, and you can localize the injured paw and create a sciatic functional index. The ATF3 knockout mice take significantly longer than the wild-type animals. And again, these are just mice that have ATF3 knocked out of their cholinergic motor neurons. Their sensory function is largely similar to the ATF3 wild-type animals, which are the red triangle and the knockout, conditional knockouts, and the circles are the wild types. So we are, we're very interested now in following up the differences between and similarities and differences between these injury programs. And with that, I'm just going to close with a few take-home points that I think may be clear from the talk, that single nucleus RNA sequencing really helps characterize cell type specific injury gene expression changes from hours to months. That these profound transcriptional reprogramming is observed across neuronal cell types after axonal injury. And ATF3 is really critical for this injury-induced reprogramming in both the peripheral and central nervous system. And as I mentioned before, the key next questions that we're addressing are, how do these injury-induced transcription factors actually orchestrate this reprogramming? Which other transcription factors are involved? And precisely how does this program get turned on epigenomically? And where are these injured state cells and transcripts in situ? And I'm going to tease with one extra slide or a couple extra slides. Those of you who have been focusing on single cell sequencing know for sure that when you dissociate all of these cells, you lose their position and where in the cell these transcripts actually are. And we've been only looking at nuclei, so that is even more limiting. And so spatial transcriptomics methods have really evolved over the last year. And so our department has been really, with Paolo Cadino and Charles Jennings and Jeff Moffett here at Harvard, have been building a MERFISH rig in our lab. Actually, it's in a shared facility outside our lab, but it's so close that we're very excited about using this very soon. And what they have here is a high power microscope connected to a fluidics chamber that allows them to image between 1 and 10,000 transcripts at a time per cell, and then their own compute cluster to analyze and store all this data. And just to give you a flavor for the type of data this can generate, in an individual neuron, we can measure 1 to 10, each one of these colors being an individual transcript. We can not only measure 1 to 10,000 transcripts, we can know its exact position along an axon or dendrite, depending on what cell you're looking at. And here's just a zoom in image, and we can end up looking at what Jeff's group has found is that some of these transcripts are localized very specifically to different parts of the axon or soma. And we're very interested now in pursuing actually the position of these transcripts after injury, both in vitro and with 3D reconstruction in vivo. And so with that, I just want to acknowledge some of the key people that have been involved in this process. Yvonne Tuchisky is a former postdoctoral fellow with Clifford Wolf, who is kind of my partner in crime in getting a lot of these studies up and running, and is now at Regeneron. And Light Yang is a research associate in my lab, and he has been just instrumental in both building the PaintSeq website, a lot of the analysis, and now is basically functioning as a senior grad student in the lab. And that should come of great intrigue to those of you who are involved in graduate school admissions, because he is currently applying, and I will be very jealous of whoever takes them into their group. Obviously, Clifford Wolf, my postdoc advisor, Mike Greenberg, So and Jakar, who is working on the motor neuron project now, Inge Chang, who made the ATF3 conditional mouse, and Emmy Lee, who really helped pioneer some of the protocols we developed while a research assistant with me as a postdoc. And just as a last plug, we have a new lab here at the Brigham, and I'm actively hiring postdoc students and research assistants. So if you are interested in this work, please feel free to contact me. and I'm happy to take any questions now.
Thank you very much, Dr. Renthal, for an excellent presentation. We do have plenty of time for questions, so go to the box in the lower right-hand corner of your panel. We can also take some verbal questions, so if you'd like to ask the question directly, you can just raise your hand, and I'll get to you as soon as I can and unmute you. So, well, the questions are coming in. Dr. Renthal, I have one to start with. Did I understand at the end of your presentation that when you were comparing the peripheral nervous system versus the CNS, it seemed like there were a lot more transcription factors change or upregulated in the peripheral, the sensory neurons compared to the motor neurons. Is that right? Did I get that right?
So I guess I would preface this by saying that that is what I showed. So but the reason for that is almost certainly because we have more statistical power in those cells right now. So I think as we increase the number of cells we get in the motor neuron, which is admittedly, around 100 right now, I think, or maybe 150 now, those numbers will have more statistical power and be able to do that. I think when we finally publish these results, we'll make sure we sample the same number of cells between the different conditions. But just to give you a flavor for the overlap, we wanted to understand what the similarities were. I wouldn't focus so much on the differences now until we have all those controls in place.
But it did seem like it took longer for the motor neurons to regenerate. Is that true also?
Well, so no,
neurons?
It seems like the injury program that we observe is actually goes away faster in the motor neurons for reasons that are still not clear yet. And I think that that's an exciting thing we're interested in. But we're not sure yet, is this a transcriptional? So the thing that I think the key questions are, is this, because the neurons regenerate more quickly, is this because there's a different transcriptional mechanism or a different signal that terminates the transcriptional response? I mean, it's certainly motor recovery doesn't complete by 7 days. So we're thinking that probably there's some signal that precedes that may be different between the two cell types, but those are ongoing questions.
Okay. Here's a question from the audience. What cell type or types are the non-neuronal cells?
So, I mean, in our DRG model, we're able to sequence satellite glia, Schwann cells, both myelinating and non-myelinating, a variety of what are likely meningeal fibroblasts, endothelial cells, and then some immune cells. The method of prep, the single nucleus prep that we do does not enrich for immune cells, so it's not something we have a lot of sampling of, but we do see significant increases in macrophages, for instance, after injury, that's just consistent with what's been published by a variety of groups. And all those data you can see on the PainSeq website.
Yeah, I found it interesting that it didn't seem like there was a lot of change in transcription factors of the glial cells. Do you think that's because they're just continuing to do their things to support the neurons as they regenerate?
Well, there are changes that occur in glia for sure. And so I guess I should emphasize for everyone that, I mean, I'm not saying that there aren't changes that are occurring. In fact, there's been some beautiful work from Valeria Cavalli's lab recently in Nature Communications that describes some of these changes that occur in satellite glia. And so I think that, there are some really important changes that are relevant for the biology of regeneration in these other cell types. But the transformation that occurs, we don't really see. There are a couple of cells, so there are some changes that occur in Schwann cells right at the site of injury that are more profound than we see in other cell types, but it's nothing to the extent that we saw in neurons.
Okay, that's helpful. Here's another question from the audience. Is there an advantage to fact sorting the CHAP positive cells, considering single cell sequencing would separate out all the different populations?
So I'm not sure I fully understand the question, but I mean, the reason why we sorted out CHAP positive cells is because the spinal cord, they make up a minority of the cells in the spinal cord, even if you just take the ventral horn. So it's mainly filled up with a variety of inhibitory and excitatory interneurons. So I think that's the reason why we did it, is just to reduce the cost of sequencing. And even as it is, just by enriching the cholinergic motor neurons, we still only have, you can see just, hundreds of cells that are injured. So, we're not sampling, we're spending a lot of money sequencing cells that are not what we're actually interested in as it is.
Right.
So if we didn't
Right.
enrich that population, we would go bankrupt very quickly.
Okay. Here's a question. Are there commonalities between injury-induced transcriptional reprogramming and the factors expressed in early development of the PNS?
So yeah, it's a great question, and it's something that we looked at, I would say, admittedly, somewhat superficially in the paper. It looks like there are some factors that overlap for sure, but from a statistical standpoint, we didn't observe a significant overlap with the data sets that we compared it against, which were FACS-sorted, RET-positive DRG neurons over a couple of time points during development. We actually are looking at this exact question in more detail now by comparing to some more recent data sets from David Ginty's lab. But the answer to that question is still unknown.
Okay. How do you determine the set of injured genes? How did you determine the set?
Right. So in this case, we basically took we performed differential expression analysis using a variety of programs, and we ended up settling on EDGAR. And we used an FDR cutoff of less than 0.05 and a log2 fold change of greater than 1, so about a 2-fold change in difference. Now, it's certainly reasonable to say, well, what about all the other genes? Because first of all, anybody who's done any gene expression analysis knows that what you call significance is somewhat of an arbitrary cutoff. And I think that there's certainly some biology there that we have ignored at the expense of clarity. But I think we focus on kind of the greatest changes that occur just so we could clearly separate out signal from noise. But I think that there's probably a lot, especially as we increase the sequencing technology, the quality of the sequencing technology, and we're able to sample more transcripts per cells. I think we'll be able to look at lower fold change genes with greater confidence.
Okay. You mentioned immune cells before, and here's a question from the audience related to that. Are these transcriptional reprogramming networks entirely intrinsic to the injured neurons, or do you think some of these transcriptional reprogramming, some of them are also modulated by immune cells?
Oh, that's a great question, and I would love to know the answer to it. They're difficult questions to answer, but I think it has been. So I think there's some data that maybe point towards a mixture, right? So I think that the initial induction of ATF3 after axonal transection and a variety of other transcription factors is likely cell autonomous. I'm actually going to have to stretch my brain to be 100% convinced of that. I mean, so what I'm pointing towards is like you can dissociate these cells in vitro and look at their levels of ATF3, and they're sky high just from dissociating them, and there's really not a lot of other cells in the mix there. That said, there are some other cells in the mix, so maybe, is that been definitively proven? Maybe not, but certainly the calcium waves that are induced that are cell autonomous are due in proximally to the injury, and that's been seen through calcium imaging from actually Alvaro Savasi's lab and Mark Tessier-Levine, where you can actually look at the calcium wave fronts in individual axons without any other cells in the picture, and so that's likely to be cell autonomous. But I think how much of it is I think a good question. We don't have a great answer for yet.
Okay. Another question from the audience. What is the resolution of the spatial transcriptomics of the MERS? Looks like you can define subcellular regions of a neuron, but would it go down to different organelles or even suborganelle resolution?
So I think if you stained for those organelles, you could get. Oh, and actually, that's a good question. I don't know the maximum limit. It'll depend on the exact scope build, and I don't think we've spec'd out what the maximum resolution yet is. But at least in Xiaohui's microscopes, I mean, she can easily get micron, sub-micron level resolution, and within. It actually, it turns out to be very complicated for multiple reasons. So It depends also on the abundance of the transcript too. So if you have a really abundant transcript, it gets a little bit harder to differentiate the spots between individual counts. So I think, you know, the actual number is going to be tough for me to say, and it's a little bit conditional. But I think for large organelles, definitely you should be able to track that.
Okay.
But I haven't seen anybody do that yet, and so there may be some technical limitations. But certainly axon versus soma is easy to do,
Right.
and within the nucleus, they've been able to look at different transcripts within different domains of chromatin. So I mean, I think it's pretty sensitive, but there'll be some caveats depending on what gene you're looking at or panel of genes.
Okay. And I have a big picture kind of question for you. So, you know, you work with patients that have headache and so forth. Are there any applications to what you're doing here with the injury models to that type of pain, or is it just specific to, you know, injury-induced pain?
Yeah, I mean, I think that's a good question, and I think, you know, the answer is I don't know yet. I think the likelihood is the injury responses are quite different from just, I mean, and they're almost certainly different from activation of, let's say, you know, changing the temperature of the tissue, or certain toxic substances that activate TRPA1 or something like that. I mean, the way these neurons fire is dramatic. I mean, they don't even really fire after injury. It's just a giant pulse of calcium. It's a calcium wave that propagates to activate this transcriptional program. So, I mean, I think the proximal insult is going to be very different, and so I assume that the transcriptional responses will be quite different too. But there may be some overlaps that are relevant. But I think, you know, the transcriptional profiling, right, and I think relevant to what, you know, you guys are doing at AnaBios, you know, of human tissue is really critical for understanding some of the potential targets. And so that's something that, you know, in the trigeminal ganglia we've been very involved in, and certainly there's a consortium of people, of investigators like Ted Price and Rob Giroux, who are also very interested in this in DRG. And so I think, you know, this is an active issue. I mean, an active ongoing area of research that we're all trying to do.
Right. Yeah, it's very important to know the type of pain that you're going after when developing therapeutics, and I think that's one of the challenges, and one of the reasons it's been difficult, you know, in the past decade or so, is the wrong therapeutic area or population of patients was gone after with some of these compounds.
Right, and I think, you know, the animal modeling of using nerve injury as quote unquote chronic pain or neuropathic pain is one really small subset, and I think, you know, we have to be cognizant. I mean, I think just looking at you know, our data we had in the paper, looking at paclitaxel induced neuropathic pain, we used a relatively low dose of paclitaxel, but it was sufficient to cause allodynia. Yet we saw really no transcriptional evidence of this injury-induced reprogramming. So I think there's definitely an important difference between pain and injury, but there is a lot of overlap too that I think is obvious, and so dissecting out which is which is an important problem for developing new therapies.
Yep. All right. Well, again, thank you so much, Dr. Renthal. We're getting close to the top of the hour, so I just wanted to thank you for taking the time to provide this webinar, especially in this day and age without conferences. So this is much appreciated by all those who attended and registered. Would like to let everyone know that our next webinar will be January 28th. Dr. Carol Melotto from McGill University will be talking about chronic orofacial pain. Again, another different type of pain that will be the focus of that webinar. I'd like to thank our Director of Marketing, Gary Watkins, for putting together this webinar. And thank you for all attending, and hope you all have a great end of the week. Thanks, everyone.
Thanks again.
Thank you. Bye for now.